Hello friends and welcome to CEC live lectures. Friends, we know that the process of drug discovery is a dynamic one because over the period of time it has evolved. Now when I say dynamic, it doesn't mean that the essence of drug discovery process has changed. The essence of this process remains the same, but lots and lots of modifications and advancements have been done in the field of technology that is being used, the methods that are being used. And amongst various technologies and methods, high throughput screening is one of the most promising process or technique in the field of drug discovery. Though initially high throughput screening process was largely used to screen hundreds and thousands of synthetic chemical compounds, but the usage of HTS, that is high throughput screening, has now involved in the field of natural products as well. So friends, this is what I am going to discuss today, that is the role or the potential of high throughput screening for bioactive natural products. So in this lecture, first I'll just give you a brief idea that what exactly high throughput screening is, followed by a very brief overview of its historical development, that how this technique was evolved over the period of time. Then a small comparison of the traditional assays, the assays which we conventionally used in comparison to high throughput screening methods. After that, I will discuss about the types of high throughput screening and various methods that are used in high throughput screening, how we can classify those methods. A very broad overview of protocol that is being followed if you have to perform HTS, then how HTS is used or how HTS performs in the field of natural products, what are the challenges one face when he is using high throughput screening in the field of natural products, and how we can improve our results while using HTS in natural products. So coming on to what is high throughput screening or HTS. Now HTS is nothing but it's an automated process that is used to analyze, used to screen vast number of compounds for their efficacy against a specific target, or in general to test their efficacy against a specific activity by using the automated processes and assays, which eventually leads us to hits or leads. Now if we talk about hits and leads, hits can be the compounds which have exhibited activity, but they might have certain issues in terms of their absorption or toxicity, whereas leads are the molecules which have shown a very good activity and can be directly entered into the drug discovery process for further evaluation. Now coming on to the traditional screening methods, if I just give you a brief overview that how these tests look when they were performed. So normally the assay volume was around 1 ml, and therefore these traditional screening assays were carried out in test tubes. And since one compound at a time was handled in the traditional screening methods, so the process of screening was time consuming, and according to an estimate 20 to 50 compound screening was done in 1 week. And since everything was done manually, right from the sample preparation to the preparation of reagent, to the mixing of the reagent, and to the pipetting, the process was laborious, and again it was time consuming. Now over the period of time, the field of medicinal chemistry has evolved significantly, and this evolution of medicinal chemistry has increased the speed of synthesis of various compounds or drug compounds or drug molecules. Now, this speed of, or the increased speed of synthesis of newer compounds has completely outpaced the screening speed when we used traditional methods, and therefore there was a dire need of certain methods which were quick, which were fast, which were reliable and reproducible. And this need basically lays the foundation for the discovery of high throughput screening, or the development or genesis of high throughput screening. And it all started in 1984 when in Japan scientists were screening the bacterial colonies for the production of antibiotics. And we know that the traditional screening methods on agar plate or the other methods, they took a long time in order to screen the bacterial colonies. So the scientists, they basically develop a new method which they called as filter paper disc method, wherein the filter paper disc were dipped in the bacterial broths and then they were placed over the bacterial lawns. So those bacterial colonies which used to which have capability of producing antibiotic like molecules, they give a clear zones of inhibition. Now this process, when adopted by Pfizer, it gave wonderful results, and the speed of screening was significantly increased to 800 broths per week. But the major breakthrough took place in 1986 when Pfizer adopted the usage of 96 well plates. Though the multi-well plate were initially discovered by Dr. Takatsi in 1950 in Hungary, as there was a flu breakdown in Hungary, and Dr. Takatsi was getting so many samples for analysis, so he devised a technique. He developed a 72 well plate in order to simultaneously analyze the samples. And later on, during 1960, the plates 96 well plate were discovered by Sever and Dr. Crook, which were later on commercialized. But Pfizer adopted these plates in 1986. Now, by the adoption of these plates, there was a significant reduction in the total assay volume. If we look into the volume that was used in traditional assay, as we discussed, it was nearly 1 ml or more than 1 ml, but the usage of 96 well plate reduces that volume to hardly 100 microliter. Another breakthrough or I will say transition that took place in 1986 was the shifting from the natural products to the synthetic compounds. Now again, Pfizer started the screening of the synthetic compounds by dissolving them in DMSO. But initially the speed of this process was slow, because every time we have to weigh down the required amount of the test compound and then we can perform the analysis, and therefore the speed was hardly 200 compounds per week. And later on, Pfizer adopted another new technique called as flicking technique, wherein because they believe that they have to just screen the compounds. So if they want 1 milligram of the sample, and if they are getting 0.9 milligram or 1.1 milligram, that will not drastically affect the results. So they adopted a flicking technique, wherein the sample was taken on a suitable equipment and it was just flick, and the flicking was so much optimized that every time the sample was flicked, they used to get almost equal amount of the sample. And this flicking technique further increased the speed of the screening to 800 compounds per week. Pfizer then also adopted a centralized lockstep HTS method, wherein Pfizer basically joined two of its unit, one in US and in UK. So they provide same library of compounds to both the centers and ask them to screen over wider targets. So in a single go, the same library of compounds were scanned over so many targets and the results were then correlated. Though the outcome was quite useful, but later on the whole process was coming out to be costly, so again this was not used. Then during 1990s, lot of advancements took place in the field of drug discovery by the discovery of a reporter gene assay and qualitative PCR technique. By 1992, it was found that the molecules which were in the drug discovery process, 40% of those molecules were identified through the high throughput screening. And in the later first half of the 1990s, newer applications of HTS were evolved, and now HTS was used for the ADMET, to study the ADMET characters of the compound, that is absorption, distribution, excretion, metabolism and toxicology. So, because many a times it happens that drug shows a very good efficacy, and when that drug passes all those preclinical and clinical trials and enters into the market, it after some time it exhibits toxicological reactions, and eventually the drug has to be withdrawn from the market. So, HTS is now being used to simultaneously analyze these absorption, metabolism, excretion and toxicological characters simultaneously with its evaluation of its potential for the biological efficacy. Then in 1997, further advancements took place in terms of equipment, because LC became an integral part, liquid chromatography became an integral part of HTS process, and since medicinal chemistry was synthesizing so many compounds of different chemistries, so the HPLC columns were evolving during this phase. Lots and lots of column chemistries were developed for fast throughput implementation. Then 1999 found the full integration of ADMET HTS into the drug discovery cycle, which also the speed of screening the compounds along with screening of ADMET characters was significantly increased to 360 compounds per week. During 2000 to 2010, another major advancement took place in terms of the microplates. Earlier, the plates of 96 wells were used, but during 2000 and 2010, plates with higher densities like 384 well, 1536 well plates or 3456 well plates were introduced in HTS process, which further help in miniaturizing the assays and lower the screening concentrations that were used for the test compounds. And eventually this process was further expanded its arena into genomics, crystallography, material science and environmental toxicity. And during the last 15 years, if we look into the data, then HTS has become an inevitable part of drug discovery process. And during the last 15 years, newer methodologies, newer techniques have been developed, wherein the techniques like 3D cell culture. Earlier, the cells, two-dimensional cell cultures were used, but during this period, three-dimensional cell cultures methods were developed, wherein the cell was the effect of the drug on a cell can be analyzed in its normal physiological environment, so that the matrix-matrix interaction, drug-matrix interaction can also be simultaneously analyzed. Further, another important technique, that is high content screening or high content imaging or high content analysis, was also introduced, wherein the imaging of the cell, the data that was obtained by imaging of a cell was used to study the effect of the drug on certain parameters. Though the imaging was largely giving you the data, phenotypic data, like if a drug is having a positive impact on the protein synthesis, the protein was tagged by a suitable fluorophore or a fluorescent compound. So if a drug is having a positive impact on the protein synthesis, the fluorescent content or the intensity of that fluorescence will increase, and that can be captured by imaging. Now, if we look into a brief comparison of traditional versus high throughput assays, as we discussed that traditional screening was done in test tubes because of higher assay volumes, whereas high throughput screening is done in 96 well plate or even higher, more 384 or 1536 and so on and so forth. And this increase in plate density has significantly reduced the assay volume to 50 to 100 microliter or even less. And since in the traditional assays, assay volume was higher, we used to do those assays in test tubes, larger amount of the test compound was required, that is in the range of 5 to 10 milligram, which was reduced to even less than 1 milligram in case of HTS. And if we talk about the capacity of screening or the speed of screening, traditional assays have a very slow speed of screening of test compounds, that is 20 to 50 compounds per week, whereas in case of high throughput screening, we can analyze 10,000 to 1 lakh compounds in a single day. And since in traditional assays we used to handle one compound at a time, therefore in a limited period of time, the diversity, the chemical diversity of the compounds that were screened for a particular disorder was limited, whereas in case of high throughput screening, as I told you, we can handle 10,000 to 1 lakh samples or test compounds in one day. So we can have a vast chemical diversity of the chemical test chemical compounds. The sample preparation methods and whatever methods that are followed in assay were manual in case of traditional screening assay, and HTS, as the definition we discussed, is completely an automated process. Now, if we look into the types of high throughput screening, we have conventional high throughput screening, wherein the screening frequency is 10,000 to 1 lakh samples per day. Then further advancements led to to the development of another high frequency technique, which we named as ultra throughput screening technique, which can handle more than 1 lakh samples per day. Then we have virtual screening type of high throughput screening, wherein we use computational models. We see drug protein interaction. In silico screening is another name for virtual screening, wherein the drug interactions are observed on a computer monitor and those molecules are selected which exhibit a good binding to the protein or interaction to the protein, and then those selected molecules are further evaluated on various in vitro and in vivo assays. Then another type is phenotypic screening, wherein we largely deal with the cellular morphological characters, the effect of drugs on such characters, like whether the drug inhibits the proliferation of the cell, whether drug enhances the viability of a cell, whether drug enhances the production of a specific protein inside the cell. So such kind of characters are largely observed in the phenotypic screening. Now, if we want to classify various methods that are used in HTS, we can broadly classify them into non-targeted screening methods. Now, as the name non-targeted screening, that clearly indicates we don't have a specific target for a drug, but in general we will see that whether the drug molecule is active or not. How it is producing that effect, this kind of information is largely not produced by the non-targeted screening. So, in such kind of methods, we have phenotypic assays. As we discussed, they largely give you an idea about the morphological characters or morphological changes or the disintegration or damage or cell membrane integration characters through these assays. Then we have reporter gene assays, wherein we get to know about the impact of our test compound on gene expression, protein-protein interactions, and so on and so forth. Then automated patch clamp assays are there, which largely deals with the impact of drug on the ion channels that are present. Then high content imaging, as we discussed, this technique is largely used to study the impact of drugs on the morphological aspects or on the production of a specific protein inside the cell or any other compound inside the cell. Then second is the target based screening. Now, since it is a target based screening, it largely involves the study of the test drug onto a specific target, and this target could be a receptor, it could be any protein, and therefore these assay largely deals with the enzymatic assays, the impact of drug on an enzyme, ligand receptor binding studies, wherein we see whether the drug binds to a particular receptor or not, protein-protein interactions, whether there are if there is any interaction between the two proteins or any other drug binding studies. And if we look into all these studies, these target based screening assays largely are the biochemical assays. Now, since we are performing these biochemical assays, we need to study the endpoint of those assays, and these endpoints are largely analyzed by various techniques like scintillation proximity assay, which is commonly known as SPA assay, then flow induced dispersion analysis FIDA, fluorescence resonance energy transfer FRET, and TR FRET, that is time resolved fluorescence resonance energy transfer. Then we have fluorescence polarization method, amplified luminescence proximity, which is commonly known as alpha screen, and another modification of this method, alpha LISA assays. Then electrochemiluminescence assays, mass spectrophotometry based assays. So, all these techniques are used to study the endpoints of the target based studies. Now, if we talk about scintillation proximity assay, this is largely an assay which involves the usage of radio labeled substances. So, we bind a radio label to a specific molecule, and then we have a target molecule. So, we bind this radio label to a drug molecule or a target molecule, and then we have a drug molecule. So, if there is any kind of interaction, and then the light is passed through this mixture. So, the drug molecule is also bound to a substance which absorbs the light, and then when it comes in proximity of the receptor which is bound with a radioligand or a vice versa, they will emit the energy which is absorbed by the acceptor, and once that energy is absorbed by the acceptor, it will then emit light which is captured by the detectors and is considered as a signal. Now, since it involves the usage of radio labeled, it has a very limited use. Then second was flow induced dispersion analysis, which we also call it as FIDA. Now, this assay largely depends upon the size, the change in the size which eventually changes the flow. So, just imagine there are the target molecules and the test compound molecules. They are flowing in a microcapillary and through a laminar flow. Now, if there is any kind of interaction between these two molecules, that interaction will change the size of the combination, because one drug molecule is individual and the receptor molecule is individual. Now, when they have their own size, but when they combine together, that will cause a change in the size, and this change in the size disturbs the laminar flow that is eventually captured as a signal. Again, this method is not very sensitive, and its results largely depend on so many other factors. So, this again the usage of FIDA is also very limited, but the other techniques are very widely used, and we are going to discuss them ahead. We can also classify various HTS methods on the basis of the format, and on the basis of format we can broadly classify them into in vitro assays, wherein the methods or the assays are performed outside the cell, and we can the second category would be the in vivo assays, wherein the assay methods or testing methods are largely done in the entire organisms. So, friends, in the next part we are going to discuss all these in vitro and in vivo assays in detail. Thank you.